Testing the V1 saliency hypothesis by a V1 model, Part 1. This hypothesis has received a strong support by having its prediction of an unknown but strong saliency effect by an ocular singleton confirmed experimentally. It is also easy and intuitive to see that isofeature suppression in V1 can explain the strong saliency effect by a unique feature singleton in a homogeneously featured background. In this example, the unique feature is in orientation. The saliency effect is so strong that this feature singleton is said to pop out to attract attention. And this is a pop out by unique color, or by unique motion or motion direction, or by a unique eye of origin of visual inputs. Focusing on the orientation feature and iso-orientation suppression, we can also intuitively understand why this vertical bar is more salient than this vertical bar. This is because these surrounding bars have orientation quite close to vertical, so therefore this vertical bar does not fully escape the iso-orientation suppression coming from the surrounding bars. However, more subtle and complex saliency effects in these examples are not so easily understandable just by iso-orientation suppression. Often one needs to look at how V1 neurons respond to such inputs. Furthermore, what if we have an input like this one with a uniform array of bars? Now, if the iso-orientation suppression is too strong, then the mutual suppression between these bars could lead to asymmetry breaking to give such variations of non-uniform outcomes, and such outcomes will be hallucinations. Therefore, we could test this V1 saliency hypothesis further by asking whether V1 mechanisms and its neural responses could also explain a whole body of behavioral data, some of them quite non-trivial, and they have been known for decades. Now, however, it's very difficult to do experiment to observe responses from millions of V1 neurons to all kinds of non-trivial visual inputs such as these ones. Instead, we can do a substitute by building a V1 model to get simulated V1 responses to answer these questions. So we can ask, can a single neural circuit model explain both the trivial and non-trivial saliency behavior? Is this model really a model of V1, using only plausible V1 mechanisms? Is this model well behaved, such that it will not have hallucinations? Therefore, let's show a V1 model made of V1-like elements. It should resemble real V1 in its responses to physiologically tested inputs, which are less complex. This is a way to calibrate our V1 model to the real V1. We'll use this model to observe simulated V1 responses to trivial and non-trivial visual inputs and see whether these responses are consistent with saliency behavioral effects. And this model can also help us to understand how the V1 circuit computes saliency. So the V1 neurons are modeled as orientation tuned, and the minority of V1 neurons that are not orientation tuned are omitted for simplicity. And although different V1 neurons are tuned to different scales, let's focus the model on just one scale, so all the model residue fields have the same size for simplicity as a starting point. And also the model ignores color, motion, and other features for simplicity. This is because saliency behavior, such as these ones created by spatial arrangements of oriented bars, these are among the most complex and non-trivial. So this model, it has a visual input, and these are the sampling locations of visual inputs, discrete sampling location 1, 2, 3, 4, etc. Here is one sampling location, and we call it a hypercolumn. And here is a zoomed in view of this hypercolumn, and it has 12 edge or bar detectors, okay, and so one of them is colored red for visualization, and one bar detector for one preferred orientation of a V1 neuron. Yeah, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 discrete orientations in one hypercolumn. So for example, if we have a visual image like this, it has 5 bars in the image, and these are sampled by these 5 sampling locations, and in particularly sampled by these 5 bar detectors. We color them red just to visualize them. Here are some facts about neurons and their interactions in real V1. Some neurons only excite other neurons, and the other neurons only inhibit other neurons. Outputs from V1 to other brain areas, such as V2 or superior colliculus, are only from the excitatory neurons. These neurons are also called pyramidal cells or principal neurons. The inhibitory neurons are also called interneurons, since they do not project outside V1. When two V1 neurons interact by intracortical interactions, the two corresponding receptive fields may or may not overlap with each other, but they are not far from each other, since the axons from a neuron to project to other neurons, they only project within a finite distance, typically within a few millimeters on the V1 cortical surface. And the principal neurons tend to project longer distances than the interneurons do. Two nearby principal neurons can have monosynaptic excitatory interactions. Like in this diagram, we have two pyramidal cells. They excite each other, so they are excitatory interactions. These are two inhibitory interneurons. They are connected one with each to the pyramidal cells, and visual inputs coming to these two pyramidal cells, and they can send response further downstream. And so these two neurons are mutually exciting each other. Two neurons can also have Dissynaptic inhibitions, like in this diagram. Here, these two pyramidal cells, they are excitatory neurons, so they can only excite other neurons. However, they can mutually inhibit each other indirectly via these inhibitory interneurons. For instance, this one excite that cell, which then inhibit this cell. Similarly, this excite this one and inhibit this one. So these two are mutually inhibiting each other. Two V1 neurons, when their receptive fields are not too close to each other, are more likely linked by intracortical interactions when they prefer similar rather than orthogonal orientations. In addition, when their receptive fields are co-aligned so that visual inputs exciting them could be part of a smooth contour, then these two neurons could excite each other, especially when they are having weak visual inputs. Here is our V1 model schematically. Here are 3 example cell pairs. Each pair is a mutually connected principal cell and its partner interneuron. These two pairs excite each other monosynaptically, and these two pairs inhibit each other dissynaptically. Here is an example input image to this model. For example, it could be that this principal neuron prefers a bar of vertical orientation within its receptive field, and this bar for the second principal neuron, and this bar for this third principal neuron. There are, of course, many other principal neurons and their respective partner interneurons for other bars at different spatial locations and different orientations. With this input image, the V1 model's responses after these intracortical interactions are visualized above. The thickness of each bar visualizes the strength of the response from a V1 model neuron to that input bar. For example, this principal cell responding to this input bar, so the thickness of that bar is the response level of this neuron, and the response of this neuron forms part of the V1 model's output. This horizontal bar evokes the highest response, Since the neuron responding to it escapes the iso-orientation suppression from neurons preferring and responding to the vertical bars. Here is the saliency map from these V1 neural responses. The saliency value at each location is visualized by the size of the disk depicted for each location, evoking responses. So it corresponds to the corresponding neuron's response level to that location, and always the disk is the maximum response among all neurons responding to that location. For example, at this location of the input cross, there are at least 2 neurons responding, one to the vertical bar and one to the horizontal bar. This saliency value is the maximum of these 2 responses, not the sum of these 2 responses. The response to this bar depends not only on this bar's direct input strength, but also on the contextual inputs. Since the corresponding neuron interacts with the neighboring neurons, responding to the context, therefore it depends on the neural interactions this neuron has with the neighboring neurons. This is a schematic of this neural interaction pattern. This rectangle depicts the whole visual field. Here are 3 examples of interactions from 3 example neurons at the center of each pattern with their respective neighboring neurons. They are identical, these 3 patterns are identical to each other, except for a shift of their center and a rotation of each pattern to align with the preferred orientation and with the field location of the corresponding central neuron. Each pattern has a limited range. Therefore, each neuron interacts only with neighboring neurons within this range. Zooming in, this is the interaction between the central neuron at this location and other neurons. Each neuron is visualized by a bar with its preferred orientation at its receptive field location. For example, this horizontal bar depicts the central neuron at this location, and this central neuron prefers horizontal orientation. 3 bars at this location mean that there are 3 different neurons here interacting with the central neuron. One of these neurons prefers the horizontal orientation depicted by this horizontal bar. The second neuron prefers the orientation depicted by this bar, slightly rotated clockwise from horizontal. And the third neuron prefers the orientation depicted by this third bar, slightly rotated counterclockwise from horizontal. When a bar is solid, the interaction between the neurons is monosynaptic excitation. Therefore, these 5 neurons, represented by these 5 solid bars, receive monosynaptic excitation from this central neuron. When a bar is dashed, the interaction is dissynaptic inhibition. Therefore, these 3 neurons at this location receive such inhibition from the central neuron. As a whole, this central neuron preferring a horizontal bar here monosynaptically excites these principal neurons whose preferred visual inputs are more aligned with a horizontal bar here. So that, for example, between this central bar and this bar, there could be a smooth contour linking them together. And meanwhile, this central bar dissynaptically inhibits these principal neurons whose preferred visual inputs are not aligned with the central bar here. And all these surrounding bars have their orientations similar to the horizontal orientation for this central neuron. And such a qualitative pattern of interactions, sometimes called a bow tie interaction pattern, seems to be what's present in V1, as we will show later. If we restrict to just the postsynaptic neurons tuned to horizontal orientation, like this presynaptic neuron, then these are the postsynaptic neurons, colored red for visualization. Here is another schematic to illustrate how it works with this subnetwork of V1 containing only the horizontally tuned neurons. And this is a layer of parameter cells, and this is a layer of inhibitory interneurons, one-to-one matched with the principal neurons. Only this one-to-one match is indicated to avoid cluttering in this schematic. This is a presynaptic parameter cell. It projects to these neighboring parameter cells, exerting monosynaptic excitations on them. It also projects to these interneurons, which in turn inhibit their partner principal neurons, which are therefore under the dissynaptic inhibitions. From this neuron, these other neurons with a minus sign marked in them and its own partner neuron receive directly a monosynaptic excitation from it. So if we have 5 input bars in this visual image, they directly excite these 5 principal neurons. The central bar monosynaptically excites the bar to its left and its right, and dissynaptically inhibits the other 2 bars below. Responses from the parameter cells are visualized here. They are the outputs from this V1 model. Because of the interactions in this V1 model, although these 5 bars have the same input strength, they evoke different response levels. A photograph could be sampled by the V1 model neurons through their classical residual fields to give this as inputs to the V1 model, to give such a model response pattern.